Hi everyone, my name is AJ Keefe. In this presentation, I'm going to be running through the process of apoptosis in graduate level detail. If you have any questions, please comment them. And if I say anything that is incorrect, please let me know. And thanks for watching. So this is the general overview of the points I want to hit. So I will begin with how BH3 proteins promote the formation of the MOMP, or the mitochondrial outer membrane pore, the formation of the death-inducing apoptosome, APAF1 right here, the subsequent activation of caspases right here. And then I'm also going to discuss alternative pathways to apoptosis. And finally, I will talk about how the process is cleaned up by efferocytosis. So I want to recommend that everyone watching the video pauses at each new slide and just kind of reviews the information and goes over the figure before actually playing the video and playing my voice. I think that will help you pick up the material because this presentation is very packed with information. So I think it would be really helpful to pause the videos and review it beforehand. In a healthy mitochondria, we have this happy relationship between two classes of outer mitochondrial membrane proteins. We have the pro-apoptotic proteins BAC or BACs. We see BAC in this figure, and they are constitutively inhibited by a second class of protein called the BCL2 class, which is indicated by the MCL1 and BCLXL. So the BCL2 class includes BCL2, it includes BCLXL, and MCL1, and a few others. And then the other class is the BAC or the BACs. So so under normal, healthy conditions, the pro-apoptotic BAC or BACs proteins are constitutively inhibited by the BCL2 class of proteins, indicated by the blue ones. So this interaction is disrupted by these BH3 proteins that are activated by pro-apoptotic signaling, and this is seen up here. So the BH3 domain of these proteins simply binds to a hydrophobic motif formed by the BH1 through 3 domains on BCL2. So that's really important, so let me repeat that. The pro-apoptotic BH3 proteins, or pro-apoptotic BH3 domains, they bind to a motif created by BH1 through 3 domains. And so that's critically important. That is essentially what is causing apoptosis to happen. So the BH3 proteins are the ultimate manifestation of apoptotic stimuli because when they are upregulated, they bind and they disrupt this happy BCL2 BAC relationship. They do this by binding to BCL2's BH3 domain, and they steal it away from BAC. So the BH3 protein comes in, it binds to either BCL, XL, MCL, or BCL2, and it steals it away from BAC or BACs. When BAC or BACs are freed from the BCL2 proteins because this BCL3 or this BH3 protein came in, they begin oligomerizing and they form a pore in the membrane called the MOMP, or the mitochondrial outer membrane pore. So this will sound a little bit silly, but I have this relationship analogy that might help you remember this. It helped me remember it. So imagine a happy relationship between two people. They're called BCL2 and BAC, right? However, this new person enters the scene. His name is BH3, right? His name is BH3. He comes in, and BCL2 is very attracted to BH3 and decides to dump BAC. He dumps him, and then he enters a relationship with BH3. Now, BAC is very unhappy, and he goes into this deep depression, and he takes on some very self-destructive habits, like forming a pore in his mitochondrial membrane. Now, that's kind of silly, but I think that might help you remember the process because that's kind of what happens on a molecular scale. Anyways, I also want to briefly mention that there are various experimental assays that take advantage of this permeabilization of the membrane in order to measure mitochondrial viability. So a common marker of mitochondrial health is TMRE. It's a fluorescent molecule that is very selectively taken up and accumulates in mitochondria, but it's released and it's dispersed when back or backs oligomerize and they form this pore right here. It releases things like TMRE. And so by monitoring the location and the how dispersed this TMRE is, you can actually measure whether you can basically measure the health of the mitochondria and if these back molecules or the backs are forming pores in the mitochondrial membrane. In a paper published just last year, 2016, researchers Salvador et al. Finally saw and actually proved the existence of these hypothesized MOMPs. And this was big because people had not actually seen these MOMPs until this paper came out. So in panels A and B gave us our first up-close look at a MOMP. And so this is an atomic force microscopy of an artificial membrane made with a lipid composition that mirrors the composition of a mitochondrial membrane. And in panels A, we see that the membrane by itself is pretty uneventful. Nothing interesting is happening. In panel B, we see that same membrane, but after the addition of recombinant Bax proteins. So they have, at this point, clearly formed multiple holes in the membrane, and these are the MOMPs that actually form in vivo. And so in panel C, we're seeing a closer look at one of these MOMPs, and they actually appear to be really large. So these would be the Bax proteins forming a pore around after they oligomerize, and this pore is upwards of 50 nanometers in diameter, which is far large enough for pretty much any protein, I think, to actually flow through there. So it's going to be releasing everything inside of the mitochondria, and we're going to discuss in a little bit what those proteins are and what they do to a cell. Before continuing, I want to return to the BH3 proteins that were responsible for inhibiting the BCL2 proteins and allowing back and backs to oligomerize to form that pore. So I want to provide a little bit of flavor to each one because they're all unique proteins, these BH3 proteins, and they respond to different stimuli. And so I want to give each one of these a little bit of personality to help kind of create a spot for it in your mind. So with BIM, it turns out that it's bound up and sequestered at microtubules in complex with dynein. And so stimuli such as depolymerization can potentially release and activate it. BID is a normally cytoplasmic and inactive protein, but is known to be cleaved by caspase-8. And caspase-8 is activated through the extrinsic apoptosis pathway. And we'll discuss that later. So when caspase-8 cleaves BID, it moves to the mitochondria and inhibits BCL2. And this is the primary mechanism by which the extrinsic pathway of apoptosis can initiate the intrinsic pathway of apoptosis. So this BID forms the bridge between the two. Puma and Noxa are the primary BH3 proteins that are upregulated by DNA damage and P53 signaling. Then there's BMF, which is a lot like BIM in that it's sequestered in the cytoskeletal components, and it can be activated by depolymerization. So anoikis is a type of cell death caused by a loss of integrin attachment to the extracellular matrix, and its pro-apoptotic signaling is due in part to the loss of BMF from actin and myosin. And also there's BAD, an easy one to remember because it's bad for cell survival, and BAD is phosphorylated mainly by AKT or protein kinase B, and then is bound by a phosphoserine binding protein called 1433, causing inhibition. So AKT, you might remember, is a growth signaling pathway, and so it means the cell is trying to grow, and so it it phosphorylates BAD and recruits the inhibitory 1433 protein, but a lack of growth signals basically allows BAD to lose that phosphate group and become active. And this can happen by various phosphatases like the calcium-activated calcineurin or PP1. It's also important to remind everyone that the anti-apoptotic BCL2 class of proteins can also just be generally up or down regulated by transcription or by proteasomal turnover. So also the pro-apoptotic BAC and BAX proteins can also be up or down regulated by transcription. So the BH3 proteins are generally thought of as the primary fluctuating force that makes the decision in apoptosis. But don't forget that BCL2 proteins can also be up or down regulated by things like ubiquitination, which would favor the oligomerization of BAC and BAX. For example, the BCL2-like protein MCL1, which we discussed earlier, is known to be phosphorylated by the stress kinase GSK3 beta. And this phosphorylation recruits an E3 ubiquitin ligase called MULE, M-U-L-E, standing for MCL1 ubiquitin ligase E3. And this sensitizes a cell to apoptosis because it's downregulating the amount of BCL2 proteins there are. So it increases the frequency of BAC and BAX oligomerization. And also the BCL2 proteins can be phosphorylated, which modulates their activity. And no one knows exactly which ones are activating or which ones promote apoptosis or not. But just know that the BCL2 proteins, like many other proteins in the cell, can be phosphorylated, and it can maybe sensitize a cell to apoptosis, potentially by disrupting their ability to bind BAX or BAC. And so let's move on to what proteins are released when the pore is actually formed. When a MOMP is finally formed in the membrane of a mitochondria, a swarm of toxic proteins are released from the mitochondria, two of which are actually normally involved in the electron transport chain, interestingly, and those are AIF and cytochrome C. So by far the most studied and important protein that's being released from the mitochondria is cytochrome C. So this protein, as we will see in the next slide, actually triggers the formation of the wheel of death called the apoptosome, and we'll discuss that too in a second. Another very interesting protein is SMAC. It's an inhibitor of caspase inhibitors. So by the rules of double negatives, we can say that SMAC activates caspases by inhibiting the caspase inhibitor. AIF is a slightly unusual protein that appears to be capable of inducing cell death independent of MOMP formation and cytochrome C, the apoptosome, and caspases. So it's able to do this because a proportion of AIF, about 30%, is actually located on the mitochondrial outer membrane and can be activated by non-conventional apoptotic stimuli. And once activated, AIF translocates to the nucleus and begins cleaving DNA. And so we're going to discuss AIF in the context of another form of apoptosis in a couple slides. There's also HTRA. HTRA is a mitochondrial protease that targets and cleaves caspase inhibitors. So it's kind of like SMAC in that it's an inhibitor of caspase inhibitors. And finally, I also added mitochondrial DNA, which not necessarily because it's important, but because I thought it was kind of fascinating, and there's not a whole lot of research on it, so it may actually be important. I don't think anyone's completely sure about it, but basically the presence of cytosolic mitochondrial DNA, because it looks a whole lot like bacterial DNA, it can actually trigger the intracellular inflammatory pathways. For example, it can trigger the formation of the inflammasome, NRLP3, which, if activated for too long, can trigger apoptosis. So this may be an additional molecule that's released from mitochondria to promote cell death. Once cytochrome C is released from the mitochondria, it promotes the formation of a large complex called the apoptosome. The apoptosome is also called the wheel of death, and it's formed from 7 oligomerized APAF1 subunits. Cytochrome C is able to bind one of these cytosolic APAF1 monomers at its WD40 repeat domain, and it facilitates the hydrolysis of DATP, which causes these APAF1 subunits to then oligomerize. Once 7 of these APAF1 proteins have been functionally oligomerized by cytochrome C and DATP, they will have formed at the center of this entire wheel of death complex a functional CARD domain, standing for Caspase Recruitment Domain, which is exactly what it does. It recruits and activates Caspase-9, and we'll discuss that in a bit. So cytochrome C binds APAF1, facilitates DATP hydrolysis, which allows it to oligomerize, and the oligomerization forms a functional CARD domain, which allows it to recruit and activate Caspase-9. The formation of the apoptosome is actually a pretty regulated process, because once the apoptosome is formed, it will unleash the caspase cascade, which is kind of the point of no return. So I mentioned that DATP is required for the oligomerization of APAF1 proteins, but once the DATP is hydrolyzed, it actually needs to be released and exchanged for another DATP molecule. And the exchange and regeneration of the DATP is orchestrated by this HSP70. Let's see, where is it? The HSP70 Cas-FAPI complex. And when a component of this complex is mutated or missing, the apoptosome fails to form and instead forms aggregates, and it forms a non-functional aggregate complex. So a protein like FAPI has actually earned its name as a tumor suppressor because it facilitates the apoptosome's formation, and the loss of FAPI would essentially allow cancer cells to avoid the intrinsic cell death pathway because the apoptosome won't be able to form, and Caspase-9 won't be able to be activated by the apoptosome. It's also been demonstrated that physiological ranges of calcium can influence DATP and DADP exchange. Specifically, high levels or high concentrations of calcium prevent apoptosome formation, which does seem kind of at odds with other research, because generally when I think of high calcium, I equate that with cell death, but I don't really know exactly why. Cells are complicated. We also see mechanisms of modulating cytochrome C. For example, a fascinating non-canonical function of tRNA appears to be binding and inhibiting cytochrome C, which is interesting. Potassium also appears to inhibit cytochrome C. If the apoptosome does pass its regulatory steps and successfully oligomerizes, it will begin recruiting pro-caspase-9 monomers. So these are the pro-caspases because it still retains its pro domain, which is recruited to the CARD domain. So when 2 of these pro-caspase-9 proteins are coerced next to each other at the CARD domain on the apoptosome, they cleave each other into an active conformation. So the CARD domain of the apoptosome basically just serves as a platform to bring together 2 caspase-9 proteins to activate them by cleavage. Once caspase-9 is activated, by itself it's harmless, but it does activate executioner caspases. And you can tell by their name, the executioner caspases are going to be pretty deadly. So inactive procaspases 3 and 7 are cleaved and activated by caspase-9. And then they go on, they begin the final stages of apoptosis, where they begin mass cleaving proteins at DEVD sequences. DEVD. One interesting way to assay and measure this activity of the executioner caspases is with a fluorophore that is auto-inhibited. And just like the caspase-3-7 enzymes, it can be cleaved by the caspase-3 or 7 enzymes. So if the fluorophores autoinhibitory domain is separated by a DEVD linker sequence, then caspase-3-7 enzymatic activity will actually cause fluorescence by cleaving off the inhibitory domain. So I thought that was kind of a cool way to assay apoptosis, because the enzymatic activity of the executioner caspases can actually cause fluorescence by cleaving the DEVD sequence in the fluorophore and basically unleash its fluorescence. Caspase-9, like our initiator caspase, is the last. Caspase-9, our initiator caspase, is the last chance for our cell to prevent apoptosis, because once caspase-9 is activated, a caspase cascade is initiated, and the cell will probably inevitably die. So there are multiple phosphorylations that can occur on caspase-9 that are well documented, but they don't appear to be reproducibly promoting or preventing apoptosis. So I decided to instead focus on XIAP. So this XIAP protein, or X-linked inhibitor of apoptosis, has 3 critical types of domains. The most important domain is the BIR, or BIR, the baculovirus inhibitor of apoptosis. This was originally discovered in the baculovirus, which adopted the gene for its ability to prevent apoptosis, thus allowing the virus to replicate without the cell self-destructing and killing itself. Now, it's well understood that the BIR domains of various IAP proteins bind to caspases, and they're known to inhibit them. So the BIR domains can bind to something like caspase-3 or caspase-9, and it inhibits them. In some cases, the IAP proteins also polyubiquitinate caspases using their ring and UBA domains, thus directing them to the proteasome and inhibiting apoptosis. Lastly, we previously touched on the SMAC proteins. Well, the SMAC proteins are basically decoy caspases that bind to these BIR domains, and they prevent the inhibition of the caspases by the IAP proteins. So the SMAC protein is basically a suicide inhibitor of these XIAP proteins, and it binds and won't let go, and it prevents these BIR domains from inhibiting the caspases. The general importance of the XIAP protein is illustrated in diseases of XIAP deficiency, which largely manifests as an immunodeficiency disease, because many T cells actually rely on XIAP to prevent them from prematurely entering apoptosis. But because there are so many homologs of XIAP, the general phenotype is not too severe. On the other hand, many cancers will actually upregulate XIAP by gene amplification, and this prevents entrance into apoptosis by basically flooding the cell with inhibitors of caspases and prevents their activation. Caspase-8, like caspase-2 and caspase-9, is an initiator caspase. This initiator caspase is unique, however, because it's activated by extrinsic signaling, meaning other cells nearby can activate caspase-8. This process begins with 2 main ligands, and those are FAS-L and TNF, which bind to FAS and TNFR respectively. So these are pro-inflammatory cytokines that are released by T cells or macrophages to activate these death-inducing receptors. So the binding of these ligands to their receptor causes a conformational change that produces a binding site for death-inducing signaling complexes. At the FOS receptor, for example, this one, a complex called the DISC, standing for death-inducing signaling complex, forms, and it contains a CARD domain, or a caspase recruitment domain, that is specific for caspase-8, and it thus activates it. TNFR1 does something similar, but in a way that's way more complicated. So I won't get into the specifics, but basically a protein called RIP1, RIP1, is polyubiquitinated by these CIAP proteins to produce a large polyubiquitin chain. Remember that the XIAP protein contained those UBA and ring domains to mediate polyubiquitination of caspases. Well, this homolog, CIAP1 and 2, does something similar, but at the TNFR receptors, and they target RIP1. And these polyubiquitin chains are not made in such a way to recruit the proteasome. Instead, they're made in such a way to recruit various signaling proteins like NEMO. NEMO is a protein that modulates pro-survival NF-kappa beta signaling. Now, there's like 10 other regulatory proteins, and this pathways can literally promote survival or induce cell death, completely diametric outcomes, and it's ultimately based on many different factors, including things like cell type and what's going on in the nucleus and transcription, and so it's a complicated process. But just be aware that caspase-8 is the extrinsic pathway. Caspase-2 is similar to caspase-9 in that it's another initiator caspase that cleaves executioner caspases like caspase-3 and 7, but it's activated by an entirely different complex. That is, it's activated by the pitosome, and the pitosome consists of this RAID, PID, and NPM1. So caspase-2 is not only the only caspase that's localized to the nucleolus in the nucleus, although it's also found in the cytoplasm. The pathway leading to the activation of caspase-2 is much less well understood than caspase-9. Caspase-2 is similar to caspase-9 in that it's another initiator caspase that cleaves executioner caspases like caspase-3 and 7, but it's activated by an entirely different complex. That is, it's activated by the pitosome and not the apoptosome. So caspase-2 is also the only caspase localized to the nucleolus in the nucleus, although it's also found in the cytoplasm. So just because it's the only one found in the nucleolus, just remember that it's still found in the cytoplasm. So the pathway leading to the activation of caspase-2 is less well understood than caspase-9, but there are two prevailing theories as to how it's activated. So the pitosome, consisting of these two critical proteins, PID and RAID, is a well-established activating platform of caspase-2 in the nucleolus, and it was actually recently found to associate with a third protein called NPM1 to facilitate caspase-2 recruitment, autocleavage, and activation. So PID and RAID form the CARD domain, or the caspase recruitment domain, and NPM1 is somehow responsible for directing caspase-2 to the pitosome. Because recent research by Ando et al., it's just published 2017, found that caspase-2 nucleolar activation requires NPM1 and also PID. So we have these three main proteins involved in nucleolar caspase activation, PID, RAID, and NPM1. So all this pitosome is known to trigger caspase-2 activation following DNA damage. Studies have also shown that that pitosome deletion of PID or NPM1, or actually, sorry, just PID, does not actually totally prevent caspase-2 activation. So there are alternative mechanisms of activating caspase-2 following DNA damage. A recent paper by Fava, F-A-V-A et al., discovered that caspase-2 is actually responding to errors in the cell cycle. More specifically, they found that centrosome duplication was the primary activator of caspase-2, and thus some researchers believe caspase-2 is not just responding to DNA damage, but rather to cell cycle instability that could result from DNA damage. So either way, once this caspase-2 has been activated by whichever mechanism, what it does is it begins cleaving proteins like a normal caspase. But since it's in the nucleolus, one of its main targets is HDM2, or human double minute 2. HDM2 is an E3 ubiquitin ligase that constantly ubiquitinates and degrades p53. So p53, you might remember, is a potent pro-apoptotic transcription factor that is allowed to accumulate because HDM2 is being cleaved by caspase-2. So through this mechanism, caspase-2 can upregulate p53 by cleaving HDM2. And thus it can further promote apoptosis through the conventional pathway by basic p53 upregulation of BH3 proteins. Now I mentioned that although caspase-2 is the only nucleolar caspase, it's also found in the cytoplasm with other caspases. And research has found that caspase-2 expression actually spikes following endoplasmic reticulum protein misfolding. So when proteins within the ER begin misfolding, it causes an RNA endonuclease called IR1 to become activated at the surface of the ER. And IR1 cleaves specific RNAs. And it turns out that some of the RNAs that are specifically cleaved are microRNAs that are complementary to caspase-2. And so ER misfolding triggers microRNA degradation that targets caspase-2, and so that leads to the upregulation of caspase-2. So we briefly discussed caspase-2 activation in response to DNA damage, and in this slide I want to briefly cover some of the mechanisms of DNA damage signaling as it relates to caspase-2 activation and apoptosis in general. So when a cell sustains a mutation in DNA, it signals through these two kinases, ATR and ATM, these two. ATR and ATM relay their activating phosphorylations through CHK and CHK2, or CHK1 and CHK2. And these CHK1 and 2 kinases will then phosphorylate P53 at a location that prevents it from being degraded, from being ubiquitinated by HDM2. So CHK1 phosphorylation of P53 prevents P53 from getting degraded in the proteasome because it prevents it from being ubiquitinated. Furthermore, research by Ando et al. 2013 showed that ATM phosphorylations of PID at its death-inducing domain was sufficient to induce caspase-2 activation down here by ATR. And remember, caspase-2 can cleave HDM2. So not only is HDM2's ubiquitination of P53 being blocked by CHK1 and 2, but caspase-2 is cleaving and degrading HDM2. So the net result following DNA damage is the upregulation of P53. That is basically the critical event here. And P53 is a transcription factor that oligomerizes into a tetramer that upregulates PUMA and NOXA, among other BH3 proteins. So you can see that occurring here. You see P53 leading to the upregulation of PUMA, NOXA, and apparently BAX, according to this graphic. So P53 can also be transported into the cytoplasm following monoubiquitination, which is actually favored by HDM2 downregulation. So the theory as to how this happens is that the downregulation of HDM2, or decreased enzymatic activity, causes less ubiquitination of P53, but it's not completely abolished. And so monoubiquitination, as opposed to polyubiquitination, is favored and thus triggers cytoplasmic relocalization of some of the P53. So the lack of HDM2 ubiquitination is a kind of sensor for P53 export. And the export of P53 from the nucleus is contingent on a protein called CRM1, which is a nuclear export protein. So once P53 gets into the cytoplasm, it's able to directly and rapidly promote MOMP formation by binding back and backs, and it dislodges them from their anti-apoptotic BCL2 partners. So not only is P53 a powerful pro-apoptotic transcription factor, but it also does the job itself by going into the cytoplasm and directly binds to BAC and BACs and dislodges them from BCL-XL or BCL2, and thus promotes the formation of the MOMP and release of cytochrome C and Diablo, which is also SBAC, and this leads to the caspase cascade. Parthanatos is an interesting, and it's a new mechanism of apoptosis that earned its name because it functions without many of the typical characteristics and typical players of apoptosis. So Parthanatos, for example, is caspase independent and does not result in the formation of the apoptotic bodies or the conventional membrane blebbing that is seen in apoptosis. So it's believed that Parthanatos is a response to DNA damage and the overactivation of this protein called PARP1 or PARP1. PARP1 stands for poly-ADP ribose polymerase, and that is what it does. It polymerizes ADP ribose units onto a substrate, and so it's actually a type of post-translational modification called PARylation. And it turns out that PARylation is very important for the process of DNA damage repair and functions to recruit and modify the activity of DNA repair proteins. Because you can see that PARP1 is PARylating these targets, and it can include DNA repair proteins and plays some kind of role in repairing DNA. However, it's speculated that the overactivity of PARP due to excess DNA damage can send this protein into overdrive, which causes massive PARylation of proteins. And PARylation is devastating for two reasons. First, it uses NAD as a source of ribose. You can see NAD going into the reaction. It also uses ATP to polymerize the reaction. So its overactivity is going to be consuming these two essential metabolic reagents. NAD is critical for the generation of NADH, and thus for the function of the electron transport chain, and ATP just kind of goes without saying. It's very important. Second, PAR chains are actually mobile signaling groups. So although the mechanism isn't understood right now, they do appear to translocate out of the nucleus to the mitochondria where they bind AIF, which contains a PAR binding domain. So you can see these PAR groups are somehow getting out of the nucleus, moving to the mitochondria, and they bind to AIF at a very specific PAR binding domain. And this somehow activates AIF to dissociate from the mitochondrial surface and move into the nucleus. And so if you remember from an earlier slide, AIF, or apoptosis inducing factor, is capable of inducing caspase-independent cell death. And that is what happens when it's activated by these PAR chains. That is, it translocates back to the nucleus and begins mass cleaving DNA, large-scale DNA fragmentation. And just to illustrate how important it is to control the accumulation of PAR chains, PARylation chains, we can examine what happens when we knock out the glycosylase responsible for degrading and recycling the PAR chains. And that's done by PARG right here. You can see PARG is taking the PAR chain and it's turning it back into its component parts like NAD. Without PARG, NAD gets locked up in these PAR chains made by PARP1. It causes the SERT deacetylases, which are dependent on NAD, to become inactivated. And basically the cells starve to death because they are missing the precursor to NADH. So eventually electron transport chain fails across the cell. And ultimately in animal models, when this protein, PARG, is deleted, it actually causes neurodegeneration in the animal. Before finishing with how apoptosis is resolved, I want to mention this alternative mechanism of cell death that actually begins at the ER, and it has to do with calcium. So calcium concentrations are tightly regulated in cells with cytoplasmic levels about 20,000 times lower than the extracellular matrix. And the cell does this by either pumping calcium out of the cell or by sequestering it inside of the ER, where it's actually welcomed in the ER. The release of calcium from the ER is partially triggered by BH3 proteins. So normally the ER calcium channel, IP3, you can see this right here if it's clear enough. IP3 is inhibited by BCL2 right here. But when BH3 proteins are upregulated, they can actually bind BCL2 at the ER membrane and displace it from the IP3 calcium channel receptor. And this potentially can cause calcium release. So calcium can then signal through a ton of different proteins. I'll just highlight 3 in this slide. First of all, calpains, right here. Calpains are activated. Calpains are calcium-dependent proteases that are known to cleave various cytoskeletal components like tubulin and intermediate filaments. And so they can cleave and activate the BH3 BID protein, which is normally present in the cytoplasm in an inactive form. And so BID can then go on to inhibit BCL-X to promote Bax oligomerization. Cytoplasmic calcium can also activate the phosphatase calcineurin right here. We talked about this earlier. Calcineurin is able to activate BAD by removing its phosphate, its inhibitory phosphate that caused it to bind the inhibitory 14-3-3 protein. And so by removing the phosphate right here, it's able to bind BCL-XL and induce apoptosis. Let's see. It also activates the kinase DAPK through allosteric calmodulin activation. So calmodulin is a small protein that when bound to calcium can activate various effector proteins, which includes DAPK. And DAPK, which stands for death-associated protein kinase, when it's activated, like calcium-activated calmodulin, DAPK goes on to induce cell death. It's a kinase that it's poorly understood, but it clearly promotes apoptosis, and it's believed to promote apoptosis through P53. So in contrast to calcium's pro-apoptotic activity in the cytoplasm, the loss of calcium in the ER, so what's happening up here, can also promote cell death as well. This is because many of the folding chaperones inside of the ER are calcium dependent. So when calcium levels are depleted in the ER, protein folding actually stalls, and the ER's trafficking of protein to the Golgi is stopped. And this causes the unfolded protein response to be initiated because BIP dissociates from IR1 and PERK and a couple other things. But what you need to know is that the calcium is also needed in the ER. So the movement from the ER to the cytoplasm is causing cell death in multiple ways. And so the last major piece of the calcium puzzle and how it promotes apoptosis is what it does in the mitochondria. So calcium derived from the ER or the extracellular matrix, it can overload and flood the mitochondria, and it can cause the opening of a special channel called the MPTP, or mitochondrial permeability transition pore. So MPTP. It's very important to note that the MPTP is distinctly different from the MOMP formed by Back and Bax. MPTP is a calcium-sensitive pore on the inner mitochondrial membrane that, when it's opened by calcium overload, it allows the free flow of ions and protons and water. And when it's opened, it causes massive reactive oxygen species production and osmotic dysregulation. And eventually, it leads to the MOMP formation or even membrane rupture to allow massive cytochrome C release. So it's entirely possible that BH3 protein upregulation can sensitize a cell to mitochondrial cytochrome C release by first opening calcium channels at the ER. So to summarize, BH3 proteins can act at the ER membrane to remove BCL2's inhibition of IP3 receptors, sensitizing them to other signals to potentially help them open. And then when these IP3 receptors open their channels, they lead to a flood of calcium, which activates BAD and BID, and it potentially opens the mitochondrial MPTP. And this allows ultimately cytochrome C release SMAC release, HTRA2 release, and activation of caspases. Efferocytosis is Latin for to take to the grave, and that is the final process of apoptosis, and I think it's very appropriately named. So let's quickly recap on how we got here. We had BH3 proteins that were upregulated from some apoptotic stimuli, and then they migrated over to the mitochondrial membrane. They got all cozy with BCL2, and that released back and backs to begin oligomerizing and forming a pore in the membrane of the mitochondria. That pore then releases various pro-apoptotic proteins such as XIAP inhibitor, SMAC, and the apoptosome activator, cytochrome C. The apoptosome is formed. It uses a CARD domain to bring together two caspase-9 proteins that cleave and thus activate each other. Caspase-9 then cleaves caspase-3 and 7, which then go on mass-cleaving cellular proteins, leading to the formation of apoptotic bodies, which are basically huge vesicles of cellular contents. Those contents need to be phagocytosed, or they risk popping and releasing tons of pro-inflammatory garbage into the cellular environment. So there are three main classes of players in the efferocytosis process. We have eat me signals, don't eat me signals, and then find me signals. So first we have eat me signals. They are found on the apoptotic bodies. This includes, most importantly, the phosphatidylserine. So I'll just call it PS, phosphatidylserine. It's a membrane component that, in healthy cells, is constitutively hidden in the inner mitochondrial membrane leaflet. So this is achieved by an intermembrane protein called ATP8A1 and ATP8A2 that function to flip any phosphatidylserine molecules that happen to flip themselves spontaneously. So this ATP8A1 is constantly flipping phosphatidylserine away from the outer membrane, and there's also an active process that actively flips phosphatidylserine in response to cellular stress. For example, TMEM16 is a calcium-activated scramblase. It's a scramblase that deviously moves phosphatidylserine to the outer membrane. So it's purposefully moving phosphatidylserine to the outer membrane. And there's also XKR8. It's a protein activated by caspase-3 or 7 cleavage that also begins moving phosphatidylserine to the outer leaflet of the membrane. So you have all these different mechanisms of moving phosphatidylserine to the outer leaflet or preventing it from getting there. But once it does get to the outer membrane, it becomes a receptor for various eat-me proteins. Basically, the microglia release a protein called MFGE8 right here, and GAS6. GAS6 and MFGE8. They diffuse through the cellular environment looking for exposed phosphatidylserine on the outer membrane of cells like neurons. And it binds those phosphatidylserine molecules, and then it serves as a phagocytosis-inducing ligand. Because then when the microglia come across it, they say, oh, this must be expressing phosphatidylserine because MFGE8 is found here. It then induces phagocytosis. Another eat-me signal is, curiously, calreticulin. Calreticulin right here. This is actually an ER folding chaperone, and I have personally no idea how it gets to the cell surface during stress, but it does appear to be there during stress, and it induces phagocytosis. And actually, lastly, desialylation. I'm not exactly sure how to pronounce it. Desialylation. It seems to be required in order for a neuron to be phagocytosed. So sialic acids are found on the membrane proteins, and it's normally a potent inhibitor of phagocytosis. So it prevents the binding of these cyclic E11 and cyclic E proteins. And however, when a cell loses these sialic acids because of neuraminidase, these neuraminidase enzymes that cleave away the sialic acids, it allows the interaction between Siglix and the desialylated membrane proteins, and this induces phagocytosis. So the last class of signals are CD47 is expressed on neurons, and it binds to SERP1 alpha on the microglia, and it's another "don't eat me" signal. Oftentimes, CD47 will be upregulated in cancers because it prevents phagocytosis. This is another potent "don't eat me" signal, often seen in cancer to prevent phagocytosis. So the last class of signals that are found that are these "find me" signals, and they're released from dying apoptotic cells, and they include, strangely, ATP and DNA and mitochondrial DNA as well, and sphingosine 1-phosphate. These molecules are basically, what they're doing is they're forming a concentration gradient that says where it is, and it's dying, and it attracts the microglia just as a bacteria would. I hope you guys enjoyed the presentation. I encourage you to please comment any questions you have, or if you need some clarification on a point. I love discussing apoptosis, so I'll definitely respond. I also encourage you to subscribe and comment and like the video, and I'll be posting more videos, so check those out as well. Thank you.